Hello, thank you for attending. This is Paul Hinderliter. I'm a senior scientist with Syngenta Crop Protection, and I was going to speak today about some ongoing work that we've had in the past few years about a new way of doing inhalation risk assessment, which we're calling a source-to-outcome approach for inhalation risk assessment. And today I'm going to give a brief overview of the work that we've been doing. So first, I'd like to start by acknowledging most of the collaborators on my project here, that no one does this in a vacuum. So we've had a wide range of folks involved from Syngenta and some outside expertise from the Pacific Northwest National Laboratory, Epithelix, and Charles River, and I'll be running through all of their contributions on this as we go. So what are we doing here? As a part of pesticide re-registration and actually pesticide initial registration, there's an examination of the completeness of the toxicological database, which includes inhalation assessment. Usually these requirements will include a subchronic inhalation study, which is a guideline study of at least 21 days or generally a 90-day requirement. But the EPA in recent years has been moving towards establishing alternatives, and as they put it in the response to one of our submissions, that there might be smarter testing or alternative approaches that are suitable to inform inhalation toxicity in lieu of a subchronic inhalation study. So with this option, we have gone back and redefined the risk assessment paradigm using some of the new tools that have been developed in recent years, including in vitro tools and dosimetry models. So we've called this a source-to-outcome approach, and if you think about the world of pesticides and external products, we're talking about things that are present in the environment, and we are taking the approach that we go all the way from the source, which is where have these things come from, the exposure, things like spray quality, how are humans exposed to them, dosimetry, what happens when it gets to a human, and outcome, which is when you actually get exposure, what is the toxicity and the risk and the hazard. So the conceptual model that has always been used for this type of work is that there's an inhalation exposure. It's generally done in a rodent. You calculate a human equivalent concentration and you do your risk characterization on it. So it's the traditional toxicological animal-based model for risk characterization. What we're proposing here in our reconstruction of the risk assessment process is to take our new tools and get to the human equivalent concentration and the risk characterization, but instead of using an in vivo animal system, to go back and find a more informative system, which in this case happens to be an in vitro system, combining that with newer computational tools, in this case fluid dynamic modeling to get the dosimetry, and proceeding to the human concentration in that manner, rather than trying all of the issues that are involved in traditional animal testing. So we've started with a series of compounds that are of interest to Syngenta that actually have similar types of toxicological findings, that they lead to things like squamous cell metaplasia, irritation, starting with necrotic injury in the epithelia of the respiratory tract. So we can actually then start with that as our adverse outcome pathway. So if we can catch the beginnings of the adverse outcome pathway, we can prevent the entire pathway from being activated. So there's a new suite of tools that have come available for this, and the one that we feel has been the most appropriate is a system called the Mucilaire system, where you can see in the upper right there's it's a plate-based system where human cells are collected from donors and then plated on these transwell plates, which are then, the cells are then grown for a significant length of time until they achieve sort of a native configuration, which you can see in the sort of in the central box there, that there's basal cells, mucus cells, cilia form, there's a pseudomucus layer, and it's all sitting on a base that gives you the configuration that you would have an air interface on the top and a liquid interface on the bottom as a surrogate for the blood. As you can see in the images in the lower left from the electron microscopy, the cilia they form a native configuration. There's actually movies on the Epithelix website where you can see where they filmed these systems, and you can see the cilia beating as they would in a native system. And in the lower picture, you can see that they actually have verified that you do get the full 3D respiratory epithelia that we need. So we have this system. What can we actually do with it? So there's a lot of different assays that can be run on this system. We've chosen 3 that seem the most relevant for establishing whether or not the beginnings of our AOP are present and irritation, essentially. So we're looking at measures of membrane integrity and cell damage as markers of irritation. So the 3 that we've chosen are epithelial electrical resistance, basically how tight are the junctions between the cells? Do we still have a good intact functional membrane? Lactate dehydrogenase, it is an enzyme in the cells that's released when the cells suffer cytotoxic membrane damage. So it's sort of a way of seeing whether or not the membranes of the cells are still intact. And then resveratrol metabolism, which is a fluorescent product, which gives us an indication of the metabolic competence of the cell. So are the cells intact? Are they happy? Do we still have a good membrane? And you see across the bottom, we've for the chemical that we've studied the most heavily on this, we've run a wide concentration range to get a dose response. So the same thing that you would do in an in vivo system, we've replicated in an in vitro system to be able to know where do you actually see changes. So it's not enough to have a single concentration and say, oh, there's an effect. We can actually here quantitate with our dose response where that effect actually occurs. Now, when we measured this for our test compound on here, we actually ran it with 5 individuals with our 3 different endpoints and found that actually the response, the variation between the donors was fairly small, and that actually all 3 of the endpoints that we were examining had responses that showed a response from this chemical, the signs of irritation, within the same range. So we were able to take sort of a grand mean of these, and if you noticed on the last slide the red curve, and if you've seen those outputs before, that's from the EPA's benchmark dose software, so that we were able to fit a BMDL to this, which you can then use as our point of departure. So we have an in vitro system that, with our range of doses, gives us a dose response. It's predictive of the beginning of our adverse outcome pathway, predictive of our toxicity. And in fact, we feel it's probably a better indication of toxicity than we would have gotten out of our in vivo animal study, largely, in fact, because we can't run as many concentrations. That BMDL can then be used as the human point of departure, and I'll come back to this after we go through the dosimetry, but that gives us an anchor point for the risk assessment rather than something like a no effect level from an animal in vivo study. So we've substituted the in vitro assay for the in vivo one. So coming back again to agrochemicals, one of the differences in these from traditional inhalation exposures is that these aren't true vapors. So there's been a growing body of work over recent years to examine things that are aerosolized in the atmosphere, things like diesel soot, PM10, even things like anthrax spores. So where do things that are in the atmosphere actually get to? And I just wanted to put up a quick cartoon here to give an indication of diameter is a hugely difficult thing to keep in mind as to what it means. So a 2 micron particle and a 100 micron particle are not, they're 50 times different in diameter, but that's actually, the volume is cubic of that. So these particles, when you go from a 2 to 100 micron particle, are enormously different. These are automobile-sized things compared to peas. So it's quite a different scenario. So what tools do we have to do things with these particles once we know that that's what our exposure is? So there's 2 classical tools that we've employed for this, and the first and the easier one to handle is the multi-path particle dosimetry model, also known as MPPD, which uses classical theoretically derived efficiencies to calculate deposition and clearance of aerosols in the respiratory tract. And it's got equations in there for the standard modes of delivery, things like deposition, sedimentation, impaction, diffusion. So it's a classical type of model and very accurate and very useful, and actually can do more than just rats and humans. They've issued an update recently to version 3, which can do a lot of other species. The limitation that we found for this project with MPPD is that we need more information about the upper respiratory tract for these larger particles. So MPPD was designed for smaller particles which penetrate the respiratory tract well. We're looking for the agrochemical side for what do we do with these larger things. So we've had to move into something called computational fluid dynamics to get deposition of these larger particles. So just to give a quick graphic, and I won't get into all the details of this, is that this is the simulations out of the MPPD of across the bottom different particle sizes and where they go in the respiratory tract. So the dashed lines are rats, the solid lines are humans. You see there's a bit of a species difference. As humans are bigger, they're more able to get larger particles down into their respiratory system, but not giant particles. So you see the large peak in blue there tops out around 10 microns, and things that are larger than that have a very difficult time penetrating into the lower respiratory tract, which you can see down in the red and the green curves. So CFD, and I'm going to give about a 30-second intro to this because we just don't have the time to go through this, is basically a way of chopping up a respiratory tract into thousands or tens of thousands of small elements, putting a discrete number of particles in them and watching where they go. So they're digitized systems. You can see on the right there, that dark gray system figure is a section of a human nasal cavity with the nostril at the left and going down into the middle respiratory tract on the right. So what does that actually do? And you can then, if we take a 50 micron particle, which we simulated early on as one of our potential systems, if you look on the left-hand side here, we can show that the deposition of a 50 micron particle is almost all in the vestibule, the nose, or the sort of parts of the respiratory tract that are really not true respiratory tissue. And we get about 1.5% in the lower part of the nasal cavity and not much getting down past that. And you see on the right-hand side, this is sort of like a weather map where you can see deposition at each surface element. So the reds and the hot reds and oranges are where deposition occurs. The blues are lower deposition, and the gray where you see the outline and there's no color on top of that. That's really a section of the respiratory tract where there is no deposition occurring from the simulation. We ran this across a variety of particle sizes in humans. So you go left to right from 1 to 30 microns, and you can see the change in the depositions. For a 1 micron particle, it's just dispersed everywhere. By the time you get up to 10 or 15 microns, you can see that it's starting to concentrate on the bottom of the nasal cavity. These things are really falling from gravitational sedimentation. And by the time you get down to 30, they're all sort of caught up in the nose. Some fall on the floor, and the few that make it past sort of tumble down into the larynx and pharynx. So once we have that deposition, we can then calculate from that across the different sections of the upper respiratory tract. We can calculate what the surface concentrations would be in each of those regions. That surface concentration can then be compared with the benchmark dose that we got from our in vitro model, which we can use in our calculations to, as you can see down here on the bottom of the slide, calculate a human equivalent concentration. Again, this is now a combination of in vitro modeling and fluid dynamics. We've not had to use any animals along the way to get to here. So, and to keep this brief, I'm just going to conclude here with things that we've used this source to outcome process to address in an EPA style risk assessment. It has provided us, we hope, with a route of satisfying our inhalation study requirements for registration. I've shown briefly how we can get through these tools to get to a point of departure. It also addresses some of the uncertainty factors that are present. So we've covered our database uncertainty factor by having actually examined this in an experimental system. We have a clear effect level. We don't have to rely on a no effect determination. We can actually fit a benchmark dose. It covers our interspecies factor because we now have a humanized endpoint. We don't have to worry about extrapolation from a rodent. So the most important take-home messages from this is that we can derive the human endpoint with the dosimetry and an in vitro model to provide a more accurate estimate of risk. This isn't, and I need to reinforce this, this is not a shortcut on this. This has been a significant chunk of work that if we had gone the traditional route and satisfied the study requirements with an animal study, we would have been done a lot quicker than we had been here. So this is not a shortcut. This is actually a better estimate of the risk to humans. And we've also reduced the animal usage for studies which have not been terribly informative. And as a final note, we have an EPA Science Advisory Panel scheduled tentatively for December of 2018, coming up this winter, to review this work for application in the EPA's risk assessment process for pesticides. So thank you for listening and have a nice day.